Okay, welcome back, everybody. If I could encourage you to take your seats. Thank you. We have two papers in this concurrent session, approximately 25 minutes each. I should introduce myself. I'm Ian Crute. I'm the Chief Scientist of the AHDB, which is the organisation within which the Potato Council resides. The first paper is going to be given by Dr. Bryan from the James Hutton Institute, not far from here in Dundee. Glenn's been a major player in the international effort to sequence the potato genome, quite a landmark event for this community. And he'll say something about how this major advance was achieved, but in particular how this is now going to facilitate more efficient genetic improvement of the potato crop. So over to you, Glenn.
Right, good morning, everyone, and thanks, Ian. I'd like to start by thanking the organisers for allowing me to talk about the potato genome today. I'd like to start by saying that since the potato genome was published about a year ago, most potato geneticists, and I include myself in that, have been in a state of semi-euphoria about how fantastic it's all going to be. And we've got the potato genome. Potato genetics and breeding are not particularly easy, and how this is going to change our lives. And I think now there's a growing kind of expectation, really, that we're going to actually use this information to do something useful, both in terms of doing genetics and research in potato, but also in crop improvement. And I think that's now the thing that we really have to focus on over the next few years. And one of the things I'm often asked is how long will it be before the potato genome impacts on potato breeding? And I think it's important there to think about some of the things that Mark mentioned this morning. I think some of that does depend on whether we can use GM in potato breeding, because I think if it's by doing conventional breeding and genetics alone, then the time for the genome to make an impact might be somewhat longer, maybe sort of 10 years before the impacts are manifest. So just in my talk, I'm really going to talk, first of all, I'm going to just say a few words. I know that this is not a scientific audience, so I'm going to say something about what the potato genome actually is, and also a little bit about why we decided to sequence it, and also about how we actually sequence the potato genome, and I'll try and keep it fairly simple. And then at the end of my talk, I'll talk a little bit about how we're actually using the genome, mostly at the moment in genetics, but also how we would like to, the last couple of slides about how we're actually going to start thinking about using it for breeding. So what do we actually mean by the potato genome? So the genome of any organism is its genetic material, usually DNA. It's organized into chromosomes, as I'm sure most of you know. Conventional potato varieties have 48 chromosomes in the nucleus of each cell. And this contains all the information needed for potato plants to develop and live and even die, all of the genes, of course. The total size of the potato genome is 850 million base pairs. So for those of you who remember your basic biology, that's 850 million G's, A's, T's, and C's. And that's about twice the size of the rice genome and about 25% the size of the potato genome. So this is just a slide from Hans de Jong in Wageningen University showing a chromosomal spread actually of a diploid potato genotype. So this only has 24 chromosomes, but it's a potato nonetheless. And so this is basically what we're talking about when we say we want to sequence the genome. So conventional varieties are quite unusual because they have 48 chromosomes, as I said, but they have 12 different chromosomes, and 4 of each in a normal cultivar. And this means that a potato is a tetraploid. Cultivated potato has four copies of the genome, and as I said earlier, this actually is quite a challenge for breeding and genetics. So one thing that this slide doesn't really show is that if you imagine that potato varieties have 12 different chromosomes and 4 of each, what this slide doesn't show is that each one of these chromosomes will be slightly different because potato is an outbreeder, not an inbreeder. So it contains very high levels of what we call heterozygosity. It doesn't breed true. If you self a potato variety, you get lots of different things that don't look anything like the parental variety. So to get around these issues, a lot of the genetics and the research we do uses diploid potatoes, and these would only have two of each, so you'd have 24 chromosomes. But again, they're still generally heterozygous individuals. So why would we want to sequence it? Well, as I said, doing genetics is difficult. Breeding is slow. It's said to take up to 12 years to breed a new variety. Potato traits are generally quite complex, resulting from the action of several genes. And as I said, crossing any two varieties yields a vast number of combinations. It's not like crossing two barley or wheat varieties where everything comes out the same in the first cross. So a genome sequence would really give us the locations of all the genes in the genome, and we can obviously compare this to where the traits that we work on map and where the markers that we use in mapping map, where the markers that we use map in the genome. And so this could really make things a lot easier in the future. And as I said, should help ultimately to improve the prospects for breeding. So this actually here is a picture of the Potato Genome Sequencing Consortium. So overall, 15 different countries were involved. And as our Chinese colleague said earlier this morning, China played a large role in this. This is Sanwen Huang, who was a very influential member of the consortium. And this shows most of the project leaders and the postdocs and students working on the project at a meeting in Ireland nearly 3 years ago. And this was actually quite a pivotal moment in the Potato Genome Project because a fairly sort of landmark decision was made at that meeting. So when the Potato Genome Project had started in 2005, the project had started to sequence this genotype here. So this is a Dutch diploid heterozygous genotype called RH, and this genotype had a very good genetic map, but it was highly heterozygous, and there were some problems encountered in actually putting the genome together. So at this meeting in Ireland, we decided to switch to this DM, which is also a diploid genotype, but it's completely homozygous. I mean, it is a highly inbred genotype, as I said, completely homozygous, and we thought that this would really make things easier in terms of doing the genome sequencing, and that turned out to be the case. Now, another thing that was decided at that meeting was to go towards an approach using what's called next-generation sequencing. This is actually now referred to as second-generation sequencing because there are new technologies coming out, which I'll mention in a moment. And this cartoon from a human genome blog site is really just talking about how the sort of technology's moved on, and now it's actually possible to sequence organisms much more quickly than it was possible to do, say, 10 years ago. And the technology is evolving all the time. And at this meeting in Ireland, we decided to use some of these next-generation technologies, such as things called Illumina and 454 sequencing, to sequence the DM genome. And one thing I'd like to say is that the technology is still changing, and I don't know if you can see this. This is a conventional memory stick that you all plug into your computers every day. And within the next 12 months, one of the companies that engages in this type of technology will release a sequencer that is based on this format. So you will plug this into your computer, you will drop a piece of, drop some DNA or blood onto this thing, and it will sequence about 150 megabases of DNA in several hours. And so this is how the technology is actually changing at the moment. Very exciting times. So as I said, we sequence the DM genome using what we call a whole genome shotgun approach. And what that basically entails is just smashing up the DNA of DM into small pieces and sequencing it. And at the end of the day, we generated enough sequence data to sequence the genome more than 125 times. And this data was assembled, what we call assembled, to make larger fragments. I'll say something about that in a moment. And then these longer sequences, which we refer to as scaffolds or super scaffolds, depending how big they are, were analyzed to actually find the genes. And this is a computationally quite intense process, which I don't have time to go into. And the idea there is to actually find the pieces of the genome that actually code for proteins, enzymes, and the actual, the parts of the genome that really actually do the work. And also, I was heavily involved in leading a group to actually map these fragments back to the genetic map so we can actually reassemble the 12 different potato chromosomes. So just a bit about whole genome shotgun sequencing. So the panel at the top, when you sequence in this manner, all you can actually do is obtain the DNA sequence. This is the G's, A's, T's, and C's from the ends of these small fragments that you produce. And what you do is you produce different sizes of these fragments so that to help what we call the scaffolding process. And using this data, then what you do is you start, again, it's a computationally intense process. You look for overlaps between the different sequence reads, and then you start to build up these small fragments into larger ones, and then these kind of orange ones here are longer fragments that you sequence the ends off, and they're very important in this process. And so you gradually, like a big puzzle, you put together the genome, and end up with these longer scaffolds and super scaffolds. And at the current time, the ideal scenario would be for the potato genome to be in 12 pieces, but that rarely happens. So currently, more than 95% of the genome is represented by 400 of these super scaffolds, which is actually pretty good, and that's where we are at the moment. So what do we learn from this? That first of all, that the potato genome contains something like 39,000 genes. So this is again the coding part of the genome, and from doing various expression kinds of analysis, we think roughly 2/3 of these genes are expressed in a set of about 30 different tissues that we've looked at. That's not to say that the other 35% aren't expressed. We haven't picked up expression of these genes in the tissues that we've sampled. We know the positions of more than 95% of these genes almost exactly, so that's a big benefit. It looks as if you take the size of the chromosomes into consideration, there are roughly the same numbers of genes on each chromosome. And one of the interesting observations was that a large part of the potato genome, in common with other plants that have been sequenced, are duplicated. So they've been through this process of genome duplication in the distant past, and also that parts of the genome are actually very similar to other plants, and I'll illustrate that in a moment. And these duplications probably occurred somewhere between 60 and 100 million years ago. So we can represent the genome in this nice figure from our paper. You saw this earlier by my Chinese colleague. And one of the what this figure shows is just the 12 potato chromosomes, almost like the hours on a clock face, and the outside bit is the cytological. So if you look down a microscope, what the chromosome would look like. And really, the only key thing I want you to take away from this is that especially if you look at chromosomes 10, 11, and 12 here, this green and black panel shows where the genes are in relation to the chromosome. So the dark segments are where the genes are, and the green segments are where the repetitive sequences are. And you can see for these chromosomes in particular, and generally over the whole map, the genes, the coding parts of the genome are distributed very much towards the ends of the chromosomes. And this is quite a common observation in plant genomes, that what we call the centromeric regions contain lots of repetitive sequences. There are some genes in there, but the genes by and large are concentrated towards the ends of the chromosomes. So going back to this point about duplication, there are something like 500 regions in the potato genome that show this highly duplicated structure. And what you can actually do is compare this with other plants. And this figure here just shows an example of just one of these regions where this segment is duplicated 3 times in potato. So you have very similar sequences in potato in 3 different locations. And this shows a comparison with the weed that you'll all find in your gardens. This is a model plant, Arabidopsis thaliana. And you can actually find these sequences in different chromosomes of the 5 Arabidopsis chromosomes, and so you can see that there's a lot of conservation, and because this genome duplication occurred between 60 and 100 million years ago, the longer kind of level of conservation over a longer distance has been scrambled between the 2 species. So although the sequences are present in both, the evidence for duplication is harder to spot because of the amount of elapsed time. And so, of course, we were all very delighted when the potato genome came out in Nature in July last year. we think this has really given potato research a big boost. And as I said, everybody's very, very enthusiastic now about using the genome in their studies on potato and hopefully applying it to breeding. So I guess the next question is, what do we do now? We have the genome. I'm a potato geneticist. I actually want, in the first instance, I want to actually use the potato genome to study a lot of the potato traits that I work on, things like dormancy, tuber shape, disease resistance, some of these things. And it's really important now that we harness the genome for these types of study. So using the genome sequence, one of the first impacts has really been to develop some nice new tools for doing research. So one of these is a thing called a microarray. So this is a tool for studying gene expression. So this is actually studying the process of how RNA, messenger RNA, is produced from genes. And you know, this is usually done by subjecting the plant to a particular stress, you know, infection by some pest or disease, or heat stress or drought stress. And we had a potato microarray before, but it actually only had something like 30 to 50% of the potato genes on it. And what we've been able to do over the last few months is redesign this microarray so that it actually contains all 39,000 potato genes. So when we do an experiment now, we can see exactly which genes are upregulated, which genes are downregulated, and we know that we've got every single gene in the genome on that microarray. So this is a really powerful tool. When we do genetics, we use what you'd refer to as molecular markers, and I guess in the past we've always been working generally in terms of sort of hundreds of genetic markers for any particular genetic map that we make. So recently there's been the development of what we call a single nucleotide polymorphism panel, or a SNP panel, which contains 8,300 loci. So it's a kind of an order of magnitude better than what we had before. And these markers are actually very, very nice, whether you work at the diploid level or the tetraploid level. You can really call the genotypes for each marker very precisely in a way that you couldn't before. So this is really going to revolutionize the way we do genetics. And even though 8,000 probably seems like a lot, we're really optimistic that over the next couple of years we can actually take that up another order of magnitude, so that when we do genetics on a particular trait in a particular cross, we might be using, say, 100,000 markers instead of nearly 10,000. And again, this is really going to impact on how well we can analyze traits. So when we do genetics in potato, the normal way to do it is to develop a population, and that could be a cross between two genotypes, or it could be what we call an association population, which is like a panel of varieties or something like this. And then what we would do, this just shows one of our diploid populations, which one we've worked on quite a lot for looking at tuber characteristics. And we would score lots of traits on this, you know, tuber shape, flesh color, all sorts of things, yield. And you know, we would measure these traits, and then we would use the markers, like as I just mentioned, to make what we call a genetic map. So this shows the genetic map of the 12 potato chromosomes from this cross. And then we bring the two types of information together to actually localize where the genes for the traits that we're interested in map. And then obviously the markers that map near those locations are the sort of markers we would use in a conventional, in, say, for example, marker-assisted breeding. So the benefit now is that when we have a genetic map, and this is something we couldn't do before, is we can actually, so this is just a cartoon of a chromosome with some markers on it. And when we localize a trait now, so whether this is disease resistance, for example, you know, Mark was talking about earlier, and what the real beauty of where we are now is that we can actually see, because these markers are all sequence-based, we can actually see them in the genome. So we know exactly where they go in the genome sequence. And so, of course, if we know that our trait map's in this region, we can look more closely at this part of the potato genome and look at the genes that have been annotated in that region of the genome and say, well, knowing what we know about the trait and knowing what we know about the genes in this region, you know, is one of them, say, you know, this orange one here, a candidate for the trait that we're interested in? And for most of the genes in the genome, we have some idea from what was called the annotation process as to what those genes are involved in. And resistance genes are a good example because resistance genes in all plant genomes so far conform to a particular structure. So they're called MB-LRR genes. And they have very well-defined domains. You know, they tend to be anywhere between sort of 3 and 5 kilobases, 3 and 5,000 bases in length. And we can spot these genes amongst the 39,000 genes. And so, again, if we were mapping, say, blight resistance, to use an example from earlier, and this gene here was one of these genes, then that would be a good candidate for that trait. So how then does the genome benefit genetics? Well, we think it's going to be much easier now to identify the actual gene responsible for any particular trait that we're interested in. And we also think, you know, wearing our sort of breeder's hats, that it should be possible to develop molecular markers diagnostic for a particular trait as well. And one of the things we've come to realize in recent months is that there are something like 400 resistance genes in the potato genome. I'll illustrate that in a moment. But I think where there's a clear hole in moving forward is we need better information about what the 39,000 genes are doing. We probably only have some idea about 50% of those genes. And so one way that traditionally has been used to assign function to a gene is to make a GM plant, not for deployment in the field, I should add, but to put it in the greenhouse and overexpress the gene and then see what happens to the phenotype of the plant. And clearly we can't do that for 20,000 genes. It's just too much work. So I think a key set of tools that we need in the future are actually ways to identify what genes are doing. And another thing that's really important is to actually resequence other genotypes of potato. You might think that's typical for someone who's just finished a grant where he was given quite a lot of money to sequence potato, and now he says he wants to sequence other types of potato. But it's really important that we actually understand the genetic variation in the potato genome. And we actually have a project now to actually sequence to a lower level, something like 300 varieties of potato. And that should be a really illuminating process. I mentioned resistance genes a moment ago. So obviously you will have all seen a field like this. This is a field in Scotland showing devastation by the late blight pathogen, Phytophthora infestans. And quite a lot of our work is involved in actually trying to map and clone resistance genes to this most destructive of pathogens. And so, as I alluded to a little while ago, one of our students, Florian Duper, really recently analyzed the potato genome to actually find all of the resistance genes in the genome. And this slide here just shows the locations of these 400-odd MB-LRR genes in the 12 chromosomes. And you'll see that in many places in the genome, these genes cluster very, very tightly in position. And that's really associated with their mode of evolution. They're thought to multiply by recombinational processes, which mean that they do tend to form these clusters. And the really interesting thing is that when we actually map functional resistances to nematodes or viruses or late blight, they do tend to map to these very strongly to these clusters, especially on chromosomes 4 and 9 and 11. And so we think this is a really powerful tool now for isolating resistance genes to any particular pathogen that we want to work on in the future. And of course, this should really make things easier in terms of developing markers for resistance traits. So just to finish off, got a couple of slides just saying how I think the genome will help. So I think a key thing is, especially for the more complex traits, such a lot of the quality traits that people in this room are interested in, I think it's important that we can identify the genetic locations of what we call QTLs, that's quantitative trait locus, with relatively large effects on the traits that we're interested in, because some of the traits are very complex. So things like fry color is probably influenced by at least 10 genes on different, often on different chromosomes, and so actually breeding this into new varieties is going to be quite difficult. So we have to sort of try and find the big players. I think it's really important that we try to start building traits of sort of industry-wide importance into our breeding material, and in this I would include especially things like potato cyst nematode and late blight resistance, and maybe also some of what we call the sustainability traits, such as water use efficiency, and nitrogen use efficiency. I think it's also really important that we use the information from the genome and what we know about which genes and which varieties to help to identify parents in breeding programs. It's another key objective, and also to ask the question as to following on from what Mark said this morning about whether this can be assisted by the use of biotechnology. I think if the potato genome is going to have an impact in the shorter term, I think whatever country you work in, I think it's really important that your industry can work together to identify the key targets, because I think it's going to be I think resources are quite stretched, and it's always going to be quite difficult if you've got 20 different traits to focus on. So I think it's clear that if we can identify the key targets, maybe 3 or 4 traits, then markers for important traits can be developed. I think we have to consider the use of GM as a component of our future strategy. I think some of the key traits, again, as Mark alluded to this morning, such as things like blight resistance, really could be fixed to some extent by use of GM. And I think it's going to be very, very difficult to keep ahead of Phytophthora if we don't use GM, because I think by the time you introgress a gene from, say, a wild species, the gene is no longer functional. And I think just to finish off, another thing that needs to be considered are use of some of these new methods, which are really, so for example, things like genomic selection, which is a way of combining all the traits together to come up with an index that you can select on. And it's really being used a lot in the animal breeding world. And I think this is something that we should consider. And also, I'd urge you to look at the website of a Dutch company called Solinta. They're actually pioneering a diploid breeding method using a combination of inbreeding and hybrid production, which I think is another method that deserves further consideration. So with that, I'll stop and thank you for your attention.
Thank you, Glenn. I think we're in reasonable time, so if there are a couple of quick questions, then I'm sure Glenn would be prepared to answer those. I'm going to stand up so I can see if anybody's got questions. Yes, down here.
Sorry, Bruce McKenzie, Syngenta. From your perspective, how do you get round, as a scientist, the public anxiety towards the GM debate around an emotive crop like potatoes, given the nature of how it breeds and whatever else, where there should be negligible risk, really?
Yeah, I mean, it's quite a difficult one. I think the potato community has to really kind of try and work together on this. And I think, I think by emphasising the fact that we would always be taking a gene, a natural gene from a potato, even if it was a wild species of potato, into a cultivated potato, then I think, that's. I think we just got to work harder at it and just work as a group, in a more concerted manner to try and educate the public. It's.
Anyone else?
On the back there.
Glenn, this is BP Singh from India. 14 countries were involved for so many years to carry out this job, and the job is well done. Now, the question is the work ahead that lies, and you have already mentioned what we have to do. Should we left it alone, or should we again require the kind of consortia we developed, and we were able to achieve this job? Because otherwise, individual countries who may not have been able to do it. But do you feel now individual countries can attempt that kind of work which is left over, identification of genes for important traits, markers, and whatnot? What's your opinion on that?
No, I don't think we'll have anything quite like the potato genome consortium again. And I think from having been involved in it for 5 or 6 years, I'm not sure my nerves could cope with being involved in such a large group for a number of years in the future. But I do think collaboration is really important. So I think we have to really pick the right partnerships. If we want to work on a certain trait such as abiotic stress, then it's quite important for us, for example, to be working with people, say in India, that have environments where we can grow material, that's and subject them to those kind of stresses. And so I think teamwork is always going to be important. Obviously, we have a very strong research community in the EU, and so I think working certainly within EU projects in the next few years, I think that could be another really important area for us to work together. But I don't think we should just keep that within Europe. I think we also have to establish partnerships elsewhere. So I think teamwork's still going to be very important.
Okay, thank you for the questions, and thanks again, Glenn.